Hello, good afternoon, everyone. So welcome to this PubMLST forum focusing on STDs. So I'm very pleased to be introducing this afternoon to you. And in my talk, what I'll be doing is just showing you some of the resources available on PubMLST and how you can use this for STD surveillance and research. This afternoon's talks in particular will be focusing on Neisseria gonorrhoeae, Treponema pallidum, subspecies pallidum, and Chlamydia trachomatis. So I thought I'd bring up this first slide really to introduce to you why STD surveillance is so important. This is an infographic which I've drawn based on data available from Public Health England. And it's really depicting the number of cases in dark blue between 2010 and 2019, the number of cases in orange in 2019 only, these triangles then depicting the sort of percentage change in these cases. And really the overall trend is that there is an increasing trend in all of these STDs in England. And this is something which is seen globally. And which cannot be attributed to kind of improved diagnostics only. So really there is a need for sort of continued surveillance of STDs. These particular three bacterial organisms are quite interesting in that they share a lot of characteristics. They have a lot of characteristics in common. In particular, they are obligate human pathogens. They all cause mucosal infections and are associated with significant morbidity. They, a lot of the time, cause asymptomatic infections, which can remain undiagnosed for long periods of time. And they're extremely well adapted for persistence in the human host, evidenced by the very long associations they've had with humans. Improved and enhanced surveillance of bacterial STDs is important as well because a number of these are developing resistance to antimicrobials. So PubMLST, there are lots of platforms actually for undertaking surveillance of lots of different bacterial species. But this afternoon we're focusing on PubMLST. And PubMLST is an online resource which allows molecular data for all of these three pathogens that we're talking about this afternoon to be deposited. PubMLST accepts single gene fragment results or draft whole genome sequence assemblies or indeed finished genomes, which can be deposited in PubMLST, then associated isolate provenance metadata, and then allows a sort of population-based annotation of these data, with which you can then perform a number of additional analyses depending on your question really and what you're wanting to do. In PubMLST at the moment, at the time of this talk, there was whole genome sequence data for nearly 10,000 Neisseria gonorrhoeae, which are publicly available. Chlamydia trachomatis, there are 468 whole genome records, and Treponema, there are 91. And really, first of all, for surveillance, what we're interested in is typing, as this helps us identify strains and then allows us to perform surveillance. And there's lots of different typing schemes for these three bacterial species that are present in PubMLST. For the gonococcus, there's three different typing schemes. There's the MLST sequence types, which was actually first developed for Neisseria meningitidis, a close relative of the gonococcus. And this indexes variation found in 7 housekeeping genes. There's NG-MAST, where we're looking at fragments of variable antigens, POR-B and TPPB. And then there's NG-STAR, which is recently developed and looks at the variability found in 7 genes that are associated with antimicrobial resistance. So any gonococcus that's deposited in the database will then have all of these different annotations associated with it, allowing a sort of fine type strain designation possible for each of these isolates. The chlamydia typing schemes, there are a number of these as well that have been developed. There's the chlamydialis scheme, which is developed for analysis across the chlamydialis genus. There's the Chlamydia trachomatis typing scheme, which is developed by Deborah Dean, and who will be speaking about this a bit later on this afternoon, which is more species specific. And then there's also the Chlamydia trachomatis subsala scheme, which looks at 5 hypervariable regions and was developed by Bjorn Herman. And finally, there's the Treponema pallidum subspecies pallidum typing scheme, which was developed by Linda Guyova, who will be also talking to us this afternoon, and is also the curator for the Treponema database. And this indexes variation in 3 chromosomal gene fragments indicated here. So So a first point of call then is undertaking typing analyses of your genomes using the sort of resources on PubMLST. Another way in which we can then look at genomes is to annotate them gene by gene, which is something we've developed extensively for both the Neisseria gonorrhoeae and the chlamydialis databases. Gene by gene annotation allows any gene of interest to be analysed across populations, between populations, within single isolates, and these genes can include those associated with antimicrobial resistance or secretion systems. So for example, chlamydia has a type 3 secretion system, which is very important for virulence. Antigens, if you're interested in vaccine research, or indeed the core and accessory genomes. And here in this short video, I'm just showing you how you can access these schemes. All of these loci are organized into these schemes as you see here. And here in this particular example, I'm showing you the scheme which contains outer membrane proteins, and particularly, for example, on PEI, and you can see it's all of the alleles that are defined for it and when these were defined. Equally, in the chlamydialis database, there's also the core genome MLST scheme, which was developed by Bart Versteeg and contains all of the genes that are core to the Chlamydia trachomatis, which you can then explore each of these individually or as a whole across a population. The core genome is particularly important because we can use this to differentiate bacterial populations, and this is particularly relevant for those bacteria which may be subject to horizontal gene transfer more than others. So core genomes, once the genes are found to be core, are defined, and we have a CGMLSD scheme, we can then assign core genome sequence types for each isolate. So this is essentially the same as an MLST scheme, except that it's across the core genome. So you can imagine that, of course, there's lots of loci that are involved in the core genome. So each isolate would have its own unique CGST really, but what we can do is then group these CGSTs using preset thresholds which consist of specific locus differences. So in this diagram here we have three core genome groups essentially of isolates. In this core genome group 1, isolate A is dissimilar to isolate B using a threshold of 300 locus differences. It's different to isolate B in 300 or fewer loci only, and similarly for isolates C and D. So within this core genome group cluster 1 here, all of these isolates will be dissimilar to another isolate within this cluster in 300 or fewer loci only. Similarly then for these ones in group 2 and these ones in group 3. However, between groups 2 and 3, for example, group 3 will be dissimilar to isolates in group 2 in more than 300 loci. So it's a way in which we can kind of form essentially core genome group clonal complexes essentially of our isolates, and we can use different thresholds of locus differences. We can go down to 2 or 5 locus differences, so these isolates which form clusters at 5 or fewer locus differences would be really highly related, whereas obviously the higher the locus differences, the threshold you go, the more distantly related they will be. So we can use that to then cluster our isolates, and in particular, so there is a core genome MLST scheme defined for Chlamydia trachomatis, and core genome groups have been defined for Chlamydia trachomatis as well. Another scheme which we've been looking at is the ribosomal MLST. Ribosomal MLST is something which has been developed and is curated by James Bray. Ribosomal genes are core in all bacteria across the bacterial domain, the prokaryotic domain in fact, and they have been found to be very good at speciating particular bacterial species, or identifying bacterial species. What I've done here in this first diagram is I've compared all of the Chlamydia trachomatis genomes that are in a database, but I've compared them this time using a ribosomal MLST scheme. So each one of these circles represents an isolate, and isolates which are similar in their ribosomal MLST schemes will form clusters. So this one in orange here have a very similar ribosomal MLST alleles. I've then labeled this by core genome group using here the threshold of 200 or fewer locus differences. So all of these isolates in orange here not only do they cluster in their ribosomal MLST alleles, but they also belong to the same core genome group at this threshold of 200 or fewer loci. Similarly then for these ones in blue here. So you can see that there's a high congruence here between ribosomal MLST and core genome group. This same tree, this is a minimum spanning tree, by the way. The same tree I've labeled using the chlamydialis MLST scheme, which was developed by Yvonne. You can see some degree of congruence, however, this orange MLST scheme is more widely distributed. I've also labelled this same tree using the Chlamydia trachomatis MLST scheme developed by Deborah Dean. So you can see this time, so it's labelled by ST MLST for Chlamydia trachomatis. So these ones here are ST39. And you can see a high level of congruence between MLST, ribosomal MLST allelic profiles and your 7 locus MLST sequence types as well. These are also associated with their particular clusters as defined in Deborah's paper. So these ones here would be the subcluster 1, which are associated with trachoma, for example. These ones here, the cluster 2, the LGV. These ones here, the cluster 3, which are the STI associated isolates. And similarly, these ones here would be cluster 1. So there's a high degree of congruence here between MLST, RMLST, and core genome group. And we can also label these using, again, the Uppsala scheme, where we see there's a little bit more diversity here because obviously these are typing more hypervariable regions. Finally, using our sort of schemes and annotations, we can start to visualize our data and detect any associations and patterns. And this is something Margaret Varga has been working on in the group. So here we have a Sankey diagram. This is data that I've extracted from the gonococci. These are the gonococcal AMR phenotypes and how they're associated with their core genome groups and NG star. You can visit this if you go to our website here, and you can interact with this data yourself. So here I've selected azithromycin resistant isolates. I can see what core genome groups are associated with, NG star, and then also what countries these were found in. And this is really a way in which we can start to explore our data and find any kind of patterns or association. Here then I'm looking at ceftriaxone resistant isolates. You can see what core genome groups they've found. So core genome group 3 here at the 300 or fewer locus threshold, what NG star they were found with, and where these were found geographically as well. So to conclude then, when we're exploiting our bacterial genome data, so PubMLST is an online repository. It's a platform that accepts multiple types of data and is available for all 3 bacterial species that we'll be looking at this afternoon. There are lots of different approaches to the analysis of whole genome sequence data, and in particular what we kind of have been developing is the gene by gene annotation, which is particularly applicable for STD pathogens, and we can use this to really forensically drill down into bacterial genomes and examine these in closer detail. We can then also extract their core genome MLST sequence types and then their core genome groups, and we can use that in association with typing schemes and then look at any patterns and associations. And this can be done using more interactive approaches to exploring whole genome sequence data and data visualization. So this just leaves me now to thank everybody who's been involved in this. Everybody, lots of people in Oxford, and also all of our collaborators who are present globally. Thank you very much.